Alright, hello class. This is going to be a review video to help you out for your test. That is tomorrow. This is some of the stuff that we went over in class today. Just in case you missed any of it, review through this video so that you can get the same information. Starting out, we're going to start out with the cell cycle. This is one of the main stages or cycles that you guys need to be familiar of that talks about the mitosis stages. So this is what leads a cell up into preparing it for mitosis and cellular division. So the first thing that we're going to talk about, the first three stages combined, if we were talking about this area here all the way around to where mitosis begins, collectively for number 41 it says the first three phases combined is called what? This is going to be interphase. So 41 is interphase. The first part of interphase starts with the gap 1 phase. So this is 42, the gap 1 phase. Number 43, for the function of the gap 1 phase, this is where the cell is going to grow and is going to continue to grow. But during the most important part of all this is that the cellular organelles are going to be replicated, preparing the cell for division. The second part, for 44, the checkpoint name at the end of the gap 1 phase, it's going to go through the checkpoint, and it's actually called the G1 checkpoint. So this is D. So the gap 1 checkpoint checks to make sure that all the organelles were copied correctly, make sure that the cell's growing accordingly to prepare it for the next phase of interphase, which is going to be the S phase. So 45 for the S phase. This is where your DNA synthesis is going to occur. In other words, that your DNA is going to be replicated for the function of this. So your cell's growing, DNA is now being replicated. This is what's preparing it for the nucleus to split into two new cells. And it creates an identical set of DNA that is going to be put into each new daughter cell. So this process is what we call is a semi-conservative process because each new daughter cell is going to have an original part of the DNA strand because of the splitting and copying. From there, we go on to 47. The third part of interphase is called the G2 phase. During this phase, it's very simple. Nothing's being replicated. However, the only thing that's happening is the cell's continuing to increase in volume. And it's doing this that way there's more cytoplasm available that it can split between the two new daughter cells. So once we get through all three stages of the interphase, it goes through a very last checkpoint for 49. This checkpoint is what's called the G2 or gap 2 checkpoint. It happens at the very end of the gap 2 phase. So this basically checks to make sure that everything was copied correctly. It makes sure that your DNA was copied correctly. It checks to make sure that the cell's grown to a specific size. Basically saying, okay, you're ready to be to go on into mitosis where you can split and divide. So from there, once it enters the mitotic stages, the very first stage is going to be prophase. So for 50 at prophase, and the function here is where the nuclear envelope is going to break down and your chromosomes are becoming visible. So in this stage specifically, your DNA becomes visible and it supercoils into the chromosomes to allow you to see the actual DNA under a microscope, very much like what you guys viewed in class under the plant and animal cells. So from there, our chromosomes start to line up in the middle. This is where 52 comes into play. This is called metaphase. So the function of metaphase is just simply that your chromosomes are lining up in the very center of the cell. And then what happens for 53, when we have 54, the next stage is going to be the anaphase. So in anaphase, the spindle fibers attach to the chromosomes, and remember that there are sister chromatids that make up the chromosomes. So spindle fibers attach to the chromosomes and it splits those chromosomes at the centromere, and it divides those sister chromatids into each new side of the cell. So this is where everything's being pulled to two new sides of the cell that prepares it for wrapping all the new DNA or replicated DNA into the nuclear envelope to make two new cells. For 55, it should be R. That's what I was just saying, that chromatids are pulled to the poles. 56 is called the telophase. So in telophase specifically, this is where everything is going to be pulled to the poles. This is where your nuclear envelope is going to start to form again around all the chromosomes or those sister chromatids. Your DNA chromatids or chromatin are going to start to uncoil. So your DNA is going to be free-floating around in the nucleus of the two new nucleuses within that cell. And then the very last phase is what we call cytokinesis for 58. This is simply the dividing of the cytoplasm into two new cells. So the cell wall is going to pinch inward, creating that burrow, and then the nuclear or cell membrane is going to grow in between them and pinch it off and have two new cells. So the main thing to identify is that mitosis specifically divides the nucleus. Cytokinesis divides the cytoplasm that is found within the cell. So the overall process of 50 to 58 is what we call mitosis. So the main thing with the cell cycle is to see that a cell spends most of its life cycle growing and reproducing the organelles and DNA to prepare it for dividing again. So approximately 75% of a cell's life cycle is spent in the first three stages called interphase. The second part you guys need to know for your test is what we call, well, it's not what we call, but it's what we've gone over, the chromosomes. So specifically what I need you to know for chromosomes is how and why it condenses and how it's prepared for mitosis. So if we start with the very first part of a chromosomes, if we think back to the pictures that we had that you guys drew in your book and on your quiz, we first had a picture that looked like this. The name of this specifically is the DNA double helix. So this is where all of your information is found that codes for everything in your body. So this is the DNA double helix. What happens is our DNA, in order to start the replication process, it wraps around specific protein structures at the same intervals that are called histones. So all the blue that you see here in this picture is what we call the histones. These histones is what helps organize our DNA and keep it all in the same strands, so that way when it supercoils, everything's still in the same order and you're not going to have a giant knot out of it. This works kind of the same way as if I were to give you a bucket of rope or a pile of rope and I told you to store it in a closet. You're not just going to take that rope and throw it in a closet. You're going to look for something to wrap it around. So this is where you're going to wrap it around some kind of a structure to help organize the rope, so that way when you go and uncoil it, you're not going to have a big mess. So as our DNA wraps around these histones at the same intervals, our DNA starts to condense and it becomes what we call our sister, well, it becomes what's called a supercoiled DNA, but in turn that's what we call chromatin. So chromatin is supercoiled DNA that's wrapped around histones and it is condensed. And then eventually what happens is it condenses even farther, and then this is what we call our sister chromatid. Okay. So the important thing is that sister chromatids is what makes up our chromosomes. So one half of our chromosome is a sister chromatid, such as here in the red, and then the green part is the other half of the sister chromatid. So keep in mind, between the red and the green, these are exact identical replicas of DNA. So the red side matches identical to the green side. This is where the DNA is semi-conservative, as each new side has part of that original DNA structure. So everything's copied here. We have our sister chromatids that make it up. And if we go through and we look at the different parts of the chromosome, the very ends of the chromosome, so all four of these ends, all have telomeres on them. So telomeres, not to be confused, but is simply like a cap. What it does is it prevents the ends of the chromosome from bonding with other chromosomes. And it's just a random sequence of nucleotides that doesn't hold any genetic information, but it keeps it and prevents it from bonding with other chromosomes. The very center where the sister chromatids are held together is called the centromere. So center and centromere, if you look, that's what that blue part's representing. There's proteins in there that help hold it all together, but this is called the centromere. The centromere is actually what splits during the metaphase process, where everything's lined up in the middle. And then that centromere splits, and then during anaphase is where your sister chromatids are being pulled to the different sides of the cells. So specifically, if you were looking at it for your mitosis stages, if I can go on to a new slide here. Maybe. There we go. So when we have our sister chromatin, here's one of our sister chromatids here, and then on the other side I have the same replica of the DNA for my other sister chromatid. So I have two identical sister chromatids here, and they're held together right here at the centromere. Okay, so that is the center. And what's going to happen during metaphase is they all line up in the very middle. So this is where they all line up at, in the very center. And then during the anaphase, our spindle fibers are going to attach to these sister chromatids, and it's going to pull these sister chromatids in opposite directions. And what it's doing by doing that is it's pulling it to opposite ends of the cell. So this is where we get our cell that has our spindle fibers through it, and we have our chromosomes or our sister chromatids that are being pulled to the opposite sides. So that is part of, or half of, our original chromosome, but that is the identical genetic information that's being shared into each new side. So this is what happens during anaphase. And then in telophase, those same sister chromatids that are on the ends of your cell, the nuclear envelope is going to start to form back around them. So here we have our cell, our spindle fibers between them, but our nuclear envelope starting to form around our sets of DNA on each side of the cell. So this is telophase. And then the very last stage which we have is cytokinesis, which is the splitting of the cytoplasm. So I have my cell, I have my two nuclei, I have my DNA that's found within the cell structure, and the cytokinesis, this is where the membrane is going to start to form between the two new cells. And in turn, what it creates is I have two new cells with my DNA and other information inside the nucleus. Now the important thing to realize is that the process doesn't stop here. Both of these two new cells here, after they've undergone cytokinesis and they have split, they're going to go back through the whole process, and they're going to start all the way back up and start growing again and preparing for further duplication through interphase. So I do want you to make the connections with how the whole cycle works and why it's important to understand how and why chromosomes condense. This should conclude the vast majority of what's going to be on your test. Just to highlight a couple other things that will be on your test, you do need to know the difference between some of the cancer cells. So when we're talking about the cancer cells, we have two different types of cancer cells. We have benign cancer cells, which basically means that it is all clumped together, means it won't spread to other areas. If it's surgically removed, that's when they'll go in and they'll just cut out that area, and it can be easily removed, or it can be treated easily. The other one is what we call is malignant. So malignant cancer is a cancer that can get into your bloodstream, and when it does that, if one of these cancer cells spreads through your bloodstream, then it becomes malignant and it spreads to other parts of the body. So this is the most fatal part of cancer, is malignant tumors or malignant cells that spread throughout your entire body. So this is how and why, if you ever are diagnosed with a tumor, you will go in, they'll take a CT scan of the tumor, and then they'll tell you to come back in a couple weeks later. And what they're doing when you're going back a couple weeks later to look at that is they'll do the same CT scan, and they're looking to make sure that the clump or tumor hasn't grown or spread into other parts of the body. So these are where some of your nuclear, it's called nuclear medicine, they'll put radioactive tracers in that are protein markers that will help track cancer cells. And this is how they can see if it's concentrated in one area or if it's spread to other areas of the body. The other thing we need to know is a little bit about stem cells. So we need to know that stem cells can come from adults and embryos. Primarily, though, they come from embryos. That is the easiest place to gather our stem cells. And then we need to know the different types of stem cells. So the main different types of stem cells is we have totipotent, there is pluripotent, and there is multipotent. So identifying these, totipotent can become any cell. They can become an organ cell, they can become skin cells, they can become bone cells, they can become neurons or hair cells, they can become any type of cell. The nice thing about totipotent cells is that they can be grown indefinitely in cultures in a lab. So this is where a lot of your stem cell research plays an ethical role of people asking questions about how the stem cells are not necessarily grown, but how they are gathered, because it does come from an embryo. Pluripotent basically are cells that have become a little bit specialized, but they can't become any type of cell. So they are more concentrated, they can become more generic, such as skin cells, they can become organ cells or soft tissue cells or muscle cells. They can become any of these, but there are certain cells that they cannot become, such as your sex cells or reproductive cells. And also some of your neurons may be already specialized. So that's what we call pluripotent. Multipotent cells are very specific. So this is very specific to it can only grow skin cells, or it can only grow heart tissue cells, or it can only grow hair cells, or it can only grow cardiac muscle cells. And same way with the neurons. So these are very specific cells. So that should conclude most of our review. I would say if you know this information, you should do very well on tomorrow's test. So good luck, everyone.